Welcome to the Harold B. Lee Library's House of Learning Lecture Series. I'm Mike Hunter, the coordinator for this year's lecture series. In the Doctrine and Covenants, section 88, verse 119, it says, "Organize yourselves and establish a house, even a house of faith, a house of learning." Through this lecture series, the Harold B. Lee Library seeks to establish a house of learning by bringing students and scholars together for a discussion of ideas. And we are privileged to have, as our featured speaker today, Professor Alan Buskirk, who is the tallest member of the chemistry and biochemistry faculty. He says he measures 6 feet and 8 inches on a good day. Professor Buskirk earned his bachelor's degree in molecular biology from BYU in 1999 and his PhD in chemistry from Harvard in 2004. He maintains a federally funded research program here at BYU studying how proteins are made in bacteria, what things can go wrong during protein synthesis, and how such problems are resolved. He also teaches biochemistry courses for graduates and undergraduates. Dr. Buskirk has a long-standing interest in the history and philosophy of science. In 2008, he taught an honors course entitled "What is Life? Scientific Metaphors for Understanding the Biological World," together with Professor David Grandy from the philosophy department. Today's lecture arises from this discussion and is entitled "Atoms, Genes, and Other Useful Fictions." And despite the provocative title, Dr. Buskirk assures me that he is committed to the following philosophical propositions. The natural world is real, the human mind can know it, and we can communicate with each other. Please join with me in welcoming Professor Buskirk.
Thank you, Mike, for the introduction and for the opportunity to speak to you today. I'm very excited to have the chance to give a broader lecture on broader topics than I usually do in biochemistry classes. Today I want to talk to you about metaphor in science and how scientists come up with models that allow them to understand the natural world. So, a very common metaphor that scientists use in explaining the stability of the natural world is the atom. And this is the idea that there are stable, fundamental, indivisible particles. And because they never change, we can use them to explain why things stay the same over time. And because we can arrange them in different ways, we can explain how things do change over time. So this is a neat philosophical answer to some problems that bothered ancient Greek thinkers. The concept of the atom has been tremendously successful in chemistry and physics in the last several hundred years. And biologists thought, hey, this is a good thing, I want some of this too. And so in the late 19th century, they came up with the concept of a gene, which was specifically created to mimic the atom, right? So genes are indivisible, stable particles, heredity, which are passed on through generations and don't change. And that's how we can explain, biological traits that remain the same over time. But you can recombine them in different ways, and that allows you to explain how some heritable traits are blended over the generations. And the concept of the gene has been tremendously successful, especially in the 20th century. But interestingly, just in the moment that both of these concepts triumph, right, in their moment of greatest utility, they unravel. And the atom, it turns out, you can smash into little tiny bits. And the gene, it turns out, really isn't stable and really doesn't do the things that is often attributed to it in the literature. So today I want to talk about where these ideas come from, how scientists use metaphors, and specifically atoms and genes. So I want to start from from the idea that in philosophy of science, there's sort of a distinction between the logic of discovery, how models are invented, how the initial insight happens, versus the logic of justification, which is how you can prove something to be the case once you have some data. You prove the model to be true. And historically, philosophy of science has tended towards a logic of justification, because this is something you can do with symbolic logic and math and so on and so forth. Very little attention has been paid to the logic of discovery. How is it that scientists come up with new ideas, new models for things they don't really understand? And obviously, this creative side of science is just as important as the analytical side. I think, in general, as a university, we do a good job of teaching analytical skills, especially in the chemistry department, right? But I don't know that we do a great job of teaching creative skills. How do you come up with these sorts of things? Now, the starting point of this is that all science is done by scientists, and scientists, it turns out, this is surprising, are people. And so people have minds, and people's minds are necessarily in their brains, in their bodies, okay? So this is a radical idea, but it turns out that science is done by embodied people. And a lot of coming up with a metaphor, coming up with a model that describes something that you don't really understand yet is related to intuition and implicit knowledge. And so I want to talk to you for a minute about implicit knowledge and what that is. So implicit knowledge is our embodied experience with the world, how we relate to the space around us, how we relate to cause, to force, to other objects. This is independent of our awareness, so we don't really know that we know this necessarily. Experts are really good at implicit knowledge. They've accumulated the experience over time, so they can say, "Oh, this is probably what's going on," and you may not be able to articulate it, but it generates intuition. So this is a quote from Polanyi. He says, "Intuition arises from our capacity to perceive coherences in things." And I love this quote because this is what the moment of discovery is usually like, where you have this flash of insight. Oh, this thing that I've been studying that I don't really understand, it's like this, and you come up with a metaphor that describes what that is. It turns out that metaphors aren't just something that you were beaten down with in high school English and you never have to think about again. There's a whole field of conceptual metaphor and cognitive science which says that the way that we understand abstract concepts like time, concepts like cause, this, even though it doesn't involve language, is directly related to our concrete experience. So we understand abstract things through our concrete experience. And these come from directly emergent concepts. So these are things which are immediate and concrete. So the way I like to think about this is everything you learned before you were 2. It turns out everything you needed to know you learned before you were 2. There are amazing studies done with babies. Anytime you talk about experiments with babies, you know you're in trouble. But these are perfectly benign. You take a baby, 6 months, say, and you put it in front of some apparatus that you're going to manipulate, and then you film the baby's eyes, and you watch how long the baby watches something, because it turns out that babies look at things they think are interesting and surprising. So then you do something crazy, right? So you reach your hand in, you have an object, and then you have something suspending the object, and so you let go of the object, and the object doesn't fall because it's suspended. The baby can't see that it's suspended because you're very clever. And the baby just stares at this thing like, why isn't it falling, right? So it turns out the babies understand gravity. They understand object permanence. They understand small numerical things. They can tell if you switch 2 objects with 3 and so forth. They can do simple arithmetic even before they're a year old. This is the kind of directly emergent concept that I'm talking about. You have this sort of sense of space, sense of objects, sense of object permanence, sense of collision. If you throw something and it hits something else, the other thing should move. These sorts of things, babies understand even before they can articulate this. So these are the kinds of concrete experience from which you generate abstract thought later on. So I thought I'd give you an example of how time, which is an abstract thought, is thought of in terms of concrete experience. So here's an example. No matter how much time passes, we will remember this day. So in this metaphor, time is, this is a pedagogical technique called audience participation, in which you say something intelligent. Time is it's passing, it's movement, right? It's a truck on the freeway, it's a river going by. Time is movement, right? What about the next one? I don't want to spend my time that way. Time is money. Yes, absolutely. Hurry, we are running out of time. Time is a resource. Excellent. Okay, we are running out of this thing. There's a certain amount of time. This is an ontological metaphor. Time is a thing. It's this resource, and it's dwindling down. That was the longest afternoon of my life, or perhaps for you, that was the longest 1-hour lecture of my life. What is time here? Yeah, it's a dimension. It's length. Okay, so we're talking about length. We're talking about objects. We're talking about money. I mean, these are all very concrete things, and that's how we're thinking of time. All right, so this is an example of how an abstract thing is understood in our minds in terms of concrete ways. Here's an example for change. The transition from the ground state to the excited state is symmetry forbidden. Now, if you've taken quantum mechanics, that makes sense, and the rest of you doesn't make any sense. But what that means is you have an electron which is going from one energy level to another energy level, right? So the way it's phrased here, the metaphor is clearly movement. The electron is going from the ground state to the excited state. Now, what's weird in quantum mechanics is it turns out that the electron doesn't actually move from here to here. It just instantaneously goes. Okay, and so this was a big problem. This metaphor of movement, change as movement, was a big problem in understanding quantum mechanics when it first got started. This is from Metaphors We Live by Lakoff and Johnson. Some really interesting cognitive science work. Okay, so specifically we're interested in metaphors in science. What do metaphors do in science? So I submit to you that metaphors do everything in science, except for all the analytical part that we teach you here at BYU. Metaphors allow you to have the initial insight that this target domain that I don't really understand maps to a source domain that I do understand. Okay, so you can make links between something I know and something I don't know. Helps you make these connections. And when I give you a metaphor, it requires your brains to engage to sort of understand the intuitive experience that you have to see if you agree with it or not. You have to do the work to make those links yourself when I give you a metaphor. Metaphors help us see the target domain in a new light, and they suggest directions for experiment. Logical positivists, logical empiricists, people doing hardcore philosophy of science in the early 20th century thought that, yeah, they're metaphors and scientists use them when they originally have the ideas, but then after that you get all the data, you reduce it to a mathematical form, and we can just talk about whether or not you've proved something right or not. But I don't think that really works. I think not only do we use metaphors in creating models, but we use metaphors when we look at our data. Data only makes sense in terms of the metaphor that you're comparing them to. And then we use metaphors when we explain science, when we explain our findings to other scientists. And then when we explain them to the broad public, we use metaphor. So metaphor is involved at every step in the scientific process. Sometimes metaphors can be very damaging. I, as was mentioned earlier, work on ribosomes in the process of translation, protein synthesis in bacteria. And I was stuck on a thing for a couple of years, not making any progress, sitting at my desk. This is when I was teaching this class with Dr. Grandy. And it came to me just in a flash. tmRNA is a hacksaw. tmRNA is this RNA molecule I work on. It doesn't matter what it does. tmRNA is a hacksaw. I realized that I had been thinking this for years previously, that the structure looked like a hacksaw. And as soon as I recognized that that's what it was, I knew it was wrong. This was really interesting to me because just sort of unconsciously I had been thinking tmRNA is a hacksaw and performing all these experiments without ever explicitly making the metaphoric link. And as soon as I did, I knew it was wrong. And since then, things have been much better because it turns out tmRNA is not a hacksaw. But metaphors just like that can be damaging not only because they're wrong, but also because they hide things. The target domain is not the source domain. The country is not the same as the map, right? And so there are things which are necessarily glossed over, and those things can be really important. So metaphors can hide as much as they uncover. And sometimes the hiding is necessary if you make any progress at all, but it can also come back to really get you in the end. The other danger of metaphors is that you might actually believe them. So it's important to recognize that metaphors are not literal descriptions. The map is not the country. As Colin Turbain says, there's a difference between using a metaphor and being used by it. There's a difference between using a model and mistaking the model for the thing modeled. The one is to make believe that something is the case, and the other is to believe it. Okay? All right. So now we need to talk about atoms. Atoms have been around for a long time. Atoms date back, well, I guess that's true. The concept of the atom has been around for a long time. The concept of the atom comes from a late 5th century BCE thinker, Leucippus and Democritus. These are Greek philosophers and scientists. And as I said before, they invented atoms to explain both the stability and change in the natural universe. So sort of competing model was Empedocles' air, water, earth, fire model that Aristotle adopts and becomes so important in medieval thought. So let me give you a feel maybe for where the idea of the atom might come from. I'm going to quote a section from Lucretius, who's several centuries later, but he's building off of their ideas. And Lucretius writes a really long poem in Latin, which I won't read to you as poetry or in Latin, called De Rerum Natura. And this, in this, he quotes some examples of how you might understand what atoms are like. Whenever the rays of the sun stream in and pour light through the dark places of the house, look, and you will see many minute particles darting about in many directions through the empty air in the light of the rays. You would do well to observe these motes which you see dancing in the sunbeams. This dancing indicates that beneath it there are hidden motions of matter which are invisible. You will see that many motes, struck by unseen blows, change their courses and are forced to move now this way and now that on all sides and in every direction. Truly, this change in the direction of all motes is caused by atoms. Okay, so immediately he's giving you this. You've had this experience before. This is what atoms are like, right? So where else does this idea of an atom come from? I think part of the reason I love this quote is that he's reasoning from a sort of macroscopic thing. Look at these particles in the air to a microscopic reasoning. This is what's going on at the invisible level that you can't see. And that's a really interesting thing and what scientists do, but surprising for 400 BCE. So what sort of metaphors involved in atoms? Well, small stable things, rocks, are stable. They're not very complicated. The more complicated something is, the more likely it is to decay. Change as movement, that's a metaphor that we have in atoms where you're moving the atoms around and recombining them in different ways. The idea that you can take parts and build up a whole. I mean, these are the sorts of concrete metaphors that you end up with in atomism. So the word atom comes from the Greek atomos, which is indivisible, something you cannot cut. Greek philosophers used to sit around arguing about whether or not you could just take something and keep cutting it in half. And one set of art philosophers would say, yeah, you can keep cutting it in half, and whenever you cut it in half, there's half left over, right? Because you're cutting it in half. But the atomists would say, no, eventually you get to a point where you can't cut it anymore, and those are atoms. So that was the ancient definition. And they are invisibly small, elementary bodies of matter. They have different shapes, different sizes, different positions, and they work in the void. So it's an absolute vacuum except for the atoms. And this allows them to move and recombine in interesting ways. This was a pretty sophisticated theory by the time we get to Lucretius. So he explained solid matter by having atoms that have Velcro on them, if you will. They fit together in ways so that they don't come apart, and that's why they're solid. Whereas liquid atoms, those are round and polished and smooth, and that's why they don't stick together so well and they flow all over the place. So they were trying to do a lot with this atomic theory. Very interesting. Okay, so Plato is the philosopher that was the most important for Christian thought in the Middle Ages. And then you have the rediscovery of Aristotle in the late Middle Ages. And both Plato and Aristotle had no use for the atomists whatsoever. Okay, so this was just completely forgotten. There was a French physicist named Gassendi who revived atomism. He had Lucretius' text published and specifically wanted to come up with the metaphysics of atoms again because he wanted some new way of explaining nature. He was tired of the old Aristotelian substances. It's important, by the way, that you recognize that the ancient atomists, atomism was a metaphysical movement. It was an idea. There was no empirical basis for it, right? So these guys weren't scientists necessarily. But as soon as Gassendi revives this idea, purely because he didn't like Aristotle, right, it becomes very useful immediately in physics. So by 1700, we have Bernoulli with a physical model of what the atom is doing. So again, atoms are in constant motion. The collisions are elastic, which means whenever they hit, they bounce off like billiard balls. The speed increases with temperature. And pressure results from collision of atoms with the wall. So that's why a gas has pressure, because the atoms are always bouncing off of everything around. And it's important, again, that this is still a metaphor. And it's far away from the actual experiments that these guys were doing. So experiments like taking a pump and pumping all the air out of something and seeing what happens, right? So if you've taken chemistry, you will remember the ideal gas law, PV-nRT, right? So they were doing things like changing the volume. If you have a gas and you smush it, technical term here, you smush the gas, then the pressure is going to go up, right? And so these are the sorts of experiments they were doing, and yet their model was atoms, right? So atoms are a nice way of explaining what's going on, but we're nowhere near seeing them or really characterize them. This is just, billiard balls bouncing around as gases. About 100 years later, Dalton came up with a description of a chemical atom. So atoms in chemistry were doing entirely different things than atoms in physics were doing. Atoms in chemistry were explaining why, when you purify something all the way down to an element, like oxygen or nitrogen or zinc or something like that, they all have the same properties. And so Dalton said that's because they're all made up of individual particles called atoms, which can't be created or destroyed, but they can be recombined in definite proportions. So if you take two hydrogen atoms and an oxygen atom, you always get water. And this was a breakthrough because now you can make chemical compounds that always have the same properties with these atoms. So atoms in physics are just bouncing around. Atoms in chemistry are explaining the properties of lead or of zinc or of oxygen and how the chemical reactivity of these elements. But they really had nothing to do with each other whatsoever until the late 19th century. And then there was this big war. There was actually a big push by philosophers to say, no, there's no such thing as the atom. And you'll see even references in the early 20th century, the so-called atomic theory. And Boltzmann, a famous physicist who did a lot of work on atoms and chemistry, took his own life because he was so upset that his atomic theory was so, rebuffed by other philosophers and scientists. But the clincher was Brownian motion. So just like these motes moving around in the air that Lucretius described 2,000 years before, Einstein predicted that at really small, at the microscopic level, if you could zoom in with a microscope, you would see particles moving around jerkily like this because of their interaction with atoms. And microscopes had progressed to the point where by this point you could see this, and there was a theory that described how this works. And in 1911, I think everybody sat down and said, okay, fine, they're atoms, I give up. So now we have atoms that explain chemistry, they explain physics, they seem to match just exactly what they're supposed to. But now we've entered the modern period, and everything sort of falls to pieces. So here's an experiment that causes trouble right from the get-go. So this is J.J. Thomson's experiment in which he discovered that the electron is a particle. And what he did was he took two metal surfaces, put a voltage across them, and then you see cathode rays, which are electrons, would leave from this metal surface and go through here and hit a fluorescent screen that you can then see the electrons impacting there. And when this was discovered, it was a ray, right? And so the Germans thought, this is a wave, because the Germans are like that. They like waves. And the English said, no, this is a particle, because the English were like that. And J.J. Thomson, of course, was English, and so he spent a lot of time worrying about how he could prove that this was a particle. And what he did was he applied either a voltage or a magnetic field across this ray, and he saw that the ray moved. Okay, now rays don't move, particles move. So the electron must be a particle, right? The electron is being attracted to the positive end or towards one of the magnetic ends. And so this shows you that the momentum of this particle is the one giving it this curve. Okay, so the electron is a particle, and everyone said, okay, we give up. You get a Nobel Prize. The electron is a particle. About the same time, Rutherford was taking radioactive decay. The alpha particles are the helium nucleus and are emitted during radioactive decay of certain elements. So he was taking these big, massive alpha particles and shining them through here through a thin film of platinum, just a few atoms thick, and then detecting it with scintillations around, all the way around, okay? And he was doing this to try and probe the structure of the atom. And what he found was that some of these alpha particles, you'd expect that they just go right through, right? Because this is a very big thing moving really fast, and it's going through this thin film. What he found is that some of them, 1 in 20,000, bounced back and hit him, or came back and hit the scintillation screen right here. So what's going on is that these alpha particles, if they hit the electron cloud, no big deal, they go right through. If the alpha particles hit the nucleus of the atom, which is this big, massive thing, but really it's a small, massive thing in the middle, then it bounces off. And sure enough, it bounced right off. I have a quote here from Rutherford. It was almost as incredible as if you had fired a 15-inch shell at a piece of tissue paper and it came back and hit you. Right? This was a big surprise. But it turns out that atoms have a nucleus. All right, now they didn't stop there, of course. I'm not going to go through all of this, but physicists have been smashing things at higher and higher energies ever since then, and now it turns out that the atom isn't just electrons and protons and neutrons. It's made up of quarks, which have names like up, down, strange, charm, top, bottom, and so forth. These could combine in different ways. These are fermions, which combine to make baryons. Also, you have leptons, which are down here. And these are bosons. These guys mediate the forces, either the strong nuclear force and the weak force and gravity and electromagnetic radiation and so forth. You'll notice a couple of weird things about this chart, besides the weird names. The mass of the up quark is 2.4 mega electron volts. Now, what on earth is an electron volt? It is the energy of one electron moving through across 1 volt potential, right? So that's a weird way to measure mass. It's an energy. So yes, these are particles, and we call this particle physics, but they're not really particles. They're chunks of energy, right? So next time someone asks you how much you weigh, you can give the answer in electron volts, and that will surprise them. So if these are particles, they're very weird particles. And this is a picture of the Hadron Collider, which is being built in Europe. I think it's finished, actually, and they're smashing these things to see what else they can find. They're looking for this Higgs boson, okay? I think the word boson is brilliant. I have a hard time taking particle physics seriously, but it really is a serious endeavor. And I guess I should say that, if the experiments with cathode rays hadn't discovered the electron, how different technology in the 20th century would have been. Who knows what they may find when they're doing this. But let's all admit that this is weird, right? This is weird. Okay, it's going to get weirder. So something about waves. If you take waves and you add them up, waves have amplitude, right? And if you add this wave and this amplitude plus that amplitude gives you a big amplitude right there. This is constructive interference. The two waves are adding, so you have higher amplitudes here. On the other hand, if you take these two waves and you add them, this is destructive interference, and so the amplitude is much reduced. Yes, you've seen something like this in your life, no doubt. So if you take light, for example, and you shine it through here, and you have 2 slits, and everything is blocked, but the light that goes through the 2 slits, light is a wave, and so it will add up, and you'll have constructive interference in these bands, and you'll have destructive interference right there, and this proves that light is a wave. Okay. So it turns out, if you do this with electrons, you get the same result. So electrons are a particle, right? J.J. Thomson just showed that. But electrons are also a wave, because if you do a diffraction of electrons, you get a pattern that looks like this, the same sort of constructive and destructive interference that you would expect from a wave. So electrons are both particles and waves. Ironically, it was J.J. Thomson's son, G.P. Thomson, who did this experiment. So dad proved it's a particle, son proved it's a wave. They both got Nobel Prizes. It's not bad if you can work that out. By showing you all of these weird standard model particle physics things and this. What I'm trying to show you is that at the smallest level, matter behaves as if it's continuous. It doesn't behave like a particle, right? And this is what makes quantum mechanics so very, very strange. There aren't very many people who understand quantum mechanics. I certainly don't. I think if you really did understand it, it would keep you up at night. It certainly kept me up at night when I was studying quantum mechanics. But this is what I mean. Look, if you keep dividing something down and you get to an atom, right, that means it's discontinuous. You can't. It's no longer a seamless thing. It's a chunk of something. But atoms take up space, right? And so what's inside the atom? Well, if you can cut the atom and there are smaller pieces, then the atom really isn't an atom after all, right? At least it's divisible. So then you get these smaller pieces. Well, what about those? Can you cut those? And if you can, then you just have an infinite regress of discontinuity, which is, possible, I suppose, but rather unattractive from a philosophical point of view. The only other option really is that inside this discontinuous atom is continuous stuff, in which case, the fundamental nature of matter is continuity rather than discontinuity. And that's sort of the way that quantum mechanics points us, in the sense that you get really strange behaviors like entanglement of photons, where one photon knows what the other photon is doing even though they're far apart in space, and they know faster than the speed of light what each other are doing. And it's just really strange. So anyway, atoms are very useful, but they are not the metaphysical atom that the Greeks came up with 2,000 years ago. We need to move on to genes. Atoms were so successful, the biologists said, hey, I want an atom of my own. And so they came up with the word gene. And this is kind of what they had in mind. You know about Mendel and Mendel's peas because you took high school biology. You have a round pea and a wrinkled pea. You cross them, and you get a round pea because round is dominant, right? Then you take two of these, and you cross those, and you get 3 round and 1 wrinkled. And you can explain this genetically by this. And the concept of gene is very useful for explaining the stability in the fact that there was some wrinkled information here which is hidden here, right? You can't see anything wrinkled here, but it pops up again later on. So there has to be something stable that carries that wrinkledness from, Grandpa P to Grandson P, right? And it doesn't show up in the parents. So genes are fundamental units of heredity. They are stable particles that generate biological traits. This is what they were invented to do around 1900. They are both law, code, and executive power. And I'm going to tell you how, in the 20th century, this concept of the gene was shown to be right. So Thomas Hunt Morgan studied flies, worked at Columbia, and he mutated the flies and then found all sorts of weird flies. And then he crossed the weird flies to other weird flies to see how the genes were related. And what he found is that genes are on chromosomes. And the way he found this is that, if you have a fly that has dumpy wings and a fly that has brown eyes, and you cross these two flies together, a recombination event might happen in the middle of this chromosome which would separate those. And so by crossing a lot of these things and measuring their phenotypes, you can figure out how closely related on the chromosome those genes are. Of course, if they assort independently, like peas, then they're on different chromosomes, right? But so you can map where genes are on chromosomes by doing this recombination. So genes are on chromosomes, 1915. In the 1940s, it was shown that it's not the protein in the chromatin structure that has the genetic information, it's the DNA. So genes are on DNA. And this is the Avery-McCarty-McLeod experiment, which you've also read about in molecular biology, no doubt. But you have some bacteria which are smooth and kill stuff, and you have some bacteria which are rough and don't kill stuff. If you take the killer bacteria, break them open, isolate their DNA, and destroy everything, destroy their protein, destroy their lipids, destroy their sugars, destroy everything but the DNA, you can take the DNA, you can put the DNA into the non-pathogenic cells, and they take it up and start expressing information on it, and they become pathogenic. So you've transformed these bacteria into something new. And this was shown in 1944 by Avery to be the DNA itself, right? So this is proof that genes are on the DNA. And of course, the real triumph of genetics was in 1953 when Watson and Crick stole a bunch of people's data and came up with a model for how the structure of DNA looks. And this is what it looks like. You have nucleotides. There are 4 of them, A, T, C, and G, and those are on one strand of DNA like this. And on the complementary strand, you also have the same 4 nucleotides, but they're arrayed in such a way that A always pairs with T, and G always pairs with C. Okay? So if you know the sequence of one strand, you can immediately know what the sequence of the other strand is. And this is brilliant because it explains to you how you can replicate this genetic information. You just separate the strands, copy them, give one to mom, or one to the old cell and one to the new cell, and off you go, right? So you can store information by which nucleotides come in which order, and then you can replicate that information also. And this is a very, very powerful thing. The gene is stable because it's a DNA molecule, okay? And the DNA molecule has information in it, and it can copy itself. And so Crick, just a few years later, came up with the Central Dogma, which is that information is stored in DNA, it is copied into messenger RNA, which is then copied, or translated, I should say, into protein. So the information goes from DNA to RNA to protein, right? And this explains how genes do stuff, because it's not enough for genes to be stable. They have to be causal. Genes make the flies have white eyes. Genes make the flies have dumpy wings. Genes make the peas round or wrinkled. This is a great way of selling your research project, right? Genes do stuff. They cause stuff. Okay, so here we have the triumph of genetics in the 20th century. And in the next few minutes, it will unravel. So, turns out that genes are no longer indivisible, stable particles, right? They're made of nucleotides. And in order for the gene to be stable, information has to be passed from one cell to the next in exactly the way that it was found. DNA doesn't copy itself. DNA is copied by proteins. One of these proteins is called DNA polymerase. I put this up to just dazzle you with purple ribbon. This is the structure of DNA polymerase. Here's a DNA strand and another DNA strand, and here's the active site of where the chemistry is taking place. This is the DNA. Here's a C that needs to be copied. There's a G that's going to come in so that you have the right GC pair. But what decides if that G is going to be incorporated or not? Well, it's DNA polymerase, right? So the enzyme is copying it, and the enzyme is responsible for the fidelity. It's responsible for the stability of the genetic information. The enzyme is responsible for copying this thing correctly. Now, it makes mistakes. About 1 in every 1,000, maybe 10,000 nucleotides it puts in the wrong one. But that's okay, because if it does, it has a proofreading activity whereby it stops and says, oops, goes back, chops off the offending mistaken nucleotide, and then starts synthesizing again. So because it can proofread, you get more fidelities, even better. So now it only makes about 1 mistake in 10 to the 7 bases that it synthesizes. So we're getting good at copying here. But that's still not enough. For genes to be stable, for the information to be passed on from mother to daughter cell, it has to be more like 1 in 10 to the 10th mistakes. So there are DNA repair mechanisms where this machine goes along the DNA and it says, wait a minute, that doesn't match. There's a GT pair, that's not right. And so it recruits some other proteins, they cut the DNA polymerase goes back and fixes this mistake. So DNA polymerase specificity, proofreading activity, mismatch repair, you only make 1 mistake in every 10 to the 10th nucleotides that you synthesize. So you're wondering, why am I talking about DNA polymerization? This is supposed to be about DNA stability. DNA itself is not a stable molecule. If you have a C base, it can react with water. Water isn't so super reactive. You drink it regularly. But it will deaminate C and give you U, which turns your GC pair into an AU pair. Or water will react with guanosine to pop off the base. And then this will just lead to the DNA being cut and disastrous consequences for the cell. Also, if you're white and you walk out into the sun will cause your thymines to dimerize, which will then block polymerization and copying of the gene. So bad stuff happens to DNA. DNA is not stable. And the information in DNA is not copied by DNA. It's copied by cellular machines, which are really complicated, which need to do it exactly right in order to maintain that genetic information. So, to sum up, why is a gene stable? The stability of a gene appears not as a starting point, but as an end product, as a result of highly orchestrated dynamic processes. So, the genetic information is stable because the cell keeps it stable, not because of any intrinsic stability of DNA, right? So it's not that DNA is a little particle of genetic goodness that doesn't ever get changed. It's because it's constantly being repaired. It's dynamics, not statics. Okay, now in the next few minutes, I hope to convince you that genes don't cause stuff. That might be a tougher sell. First of all, though, let me go back to late 19th century biology. One of the reasons why genes didn't appear earlier was that people in the 19th century didn't care so much about the stability of genetic information. They didn't care about round peas and wrinkled peas. What they wanted to know was this. If I have an egg, how on earth does it make a baby? That's unbelievable, right? I have a single cell, and now I have a baby. How did that happen? Like, that's a freakishly weird thing. And they were pretty good at this. So what they would do is, they would grow up embryos, and then they would, lop off part of the embryo and see what happened. Not to babies, but, to whatever, frogs. And so they were studying sort of the developmental processes that built up structures and tissues and human beings, right? If a chemist or a geneticist were to look at this problem, they would just say, well, there are some steps, something happens. What's important for you to realize is that genetics, as powerful as it was, shut down developmental biology. Developmental biology died because of genetics. So when Morgan got going in the 20s and everyone started doing genetics in the 30s, people stopped doing developmental biology. You couldn't ask this question. You can't ask, how does an egg become a baby? Because guess what? They have the same genes. They have exactly the same genes. Every cell in your body has the same genes. So how do you talk about this stable thing which never changes, changing, right? Because you have to, turn things on and off and make babies, right? So this was a real problem. Developmental biology died until the 1970s. And what was it that resurrected it? Well, it's that people stopped thinking about genes acting and causing stuff and started thinking about how genes are turned on and turned off. So this is a model that Jacob and Monod published in 1961, which is extremely influential. And what they were doing was they were studying how E. coli make a protein called LAC-Z, which metabolizes lactose, which is a disaccharide. Now, if you're an E. coli cell and someone gives you glucose, you're like, sweet, that's what I want. But if I don't have any glucose around, I'd rather have, if I have lactose, I can use that. I only want to express the LAC-Z protein if there's lactose around, but not if there's any glucose. And if there's no lactose around, it doesn't make any sense to make this protein. So what happens is that there's a protein called the LAC repressor, which binds upstream of the LAC-Z gene and turns it off by blocking RNA polymerase, so you never even transcribe the LAC-Z gene. So what Jacob and Monod did was say, well, genes aren't always acting. Sometimes they're being regulated. But what is doing the regulating? And this is the key point. Genes. Genes are regulating genes. So it's a vicious circle, right? Very, very interesting. So now we can talk about genes being regulated. Something else is now in control. It also raises the problem of what is a gene. In bacteria, you have these regulatory elements which are right next to the gene. In humans, these regulatory elements called enhancers can be 10 or 100 kilobases, 100,000 bases upstream of the coding sequence for the protein. You can find these regulatory elements. Are the regulatory elements part of that gene, or are they separate from the gene? I don't know. That's a tough problem. Also, it was found that when sequences in the DNA have junk DNA, if you will, in the middle of them called introns, and when the DNA gets copied into RNA, these introns, which are now light green, get cut out or spliced out like you'd splice out bad scenes from a movie. And so you end up with the protein coding pieces, which are called exons, 1, 2, and 3. But it turns out that you can take this mRNA and splice it in all kinds of different ways. You could splice it to give you 1, 2, and 3. You can splice it to give you 1 and 2 or 1 and 3. And so there are a number of different proteins that you can make from a single mRNA. Now, which is the gene? Is this the gene? Is that the gene? Is this or this the gene? What's the gene? I don't know. In Drosophila, there's a gene called DSCAM, D-S-C-A-M. It's spliced in 40,000 different ways to give you 40,000 different proteins. Which one of those is the gene? I mean, how do you even begin to answer that question? So genes become really hard to define, and scientists now just sort of generally use them in whatever way they think is best. All these organisms, yeast, flies, mice, people, have essentially the same genes, more or less. We just use them in different ways. So clearly genes aren't causing what's going on. The system is using them. So I want to leave you with a metaphor that I like. I've recently decided that I like cooking because it turns out I like eating, especially breakfast. So I like to think of genes as being a recipe. If I go to Bittman here and I open up the book and I say, I want a recipe for overnight waffles with yeast, I then follow the recipe, put all things together, and then make the waffles, right? So genes are like the recipe. Nobody would say that recipe made the waffles or the recipe caused the waffles because, I'm the one doing the making here. Now, if I change the recipe, if Bittman was playing with me and he put in, add 2 cups of sulfuric acid instead of 2 cups of milk, yeah, that would affect my waffles, right? So genes do have, the information is causally important, but really cells, the cellular system, the organism, has a developmental program, and when it needs something, it goes back in and says, okay, I need some more of this, make me some more waffles, and it uses the DNA to make some component that it's missing, right? And this is very different from the sort of selfish genetic determinism that is so popular in the press and in some scientific circles. We need to learn as biologists how to study the system. And we're getting better at looking at whole genomes, looking at all the mRNAs in a cell at once, looking at all the proteins in the cell at once to monitor the whole system at one time. But even more important, we need to learn how to think about the cellular system in terms of the sort of network analogies that come from computer science and engineering that we don't know much about. Anyway, I need to stop, but I hope I've convinced you that the stability of the natural world is not due to a fundamental particle. Okay, it's due to the interactions of things which maintain the order of the system. All right, I guess I have a few minutes for questions.